Hello, everyone. On behalf of the ESDR board, welcome to our next ESDR Kitchen Live event. My name is Sprecher, and I'm a dermatologist working at Tel Aviv. I hope you are all well. The ESDR, as you may know, has decided to make the best of these challenging times and launched a month ago a series of live events straight from the ESDR kitchen. So we will be cooking for you over the next months 4 types of dinners. Last week, we enjoyed a delicious sweet and sour meal with a debate on the pathogenesis of melanoma. But today, it's all about master chefs. And I'll now hand over to Professor Bernhard Homey, who will get you seated for our first molecular cuisine session. Bernhard.
Yeah, thank you, Ellie. My name is Bernhard Homey. I'm working at the Department of Dermatology at the University Hospital in Düsseldorf, and it's my great pleasure to introduce Professor Chris Griffiths, who will provide us with his personal insights into the clinic and pathogenesis of psoriasis. And Chris is one of the world leading psoriasis experts, and if you read his CV, you recognize that he is an impressive long-distance runner who never sleeps. So Chris was trained in London and in 1994 appointed as the foundation chair in dermatology at the University of Manchester. He held several senior positions at the University of Manchester, such as faculty research dean and the director of Manchester Academic Health Science Center. He received numerous highly prestigious awards, and Professor Griffiths is currently the president of the ESDR. His oeuvre contains 674 PubMed-listed papers. His H-index is 127. He has a career-long interest in all aspects of psoriasis, and I'm really looking forward to your presentation, Chris, and yeah, it will be great, so please. Oh, one additional thing I have to mention. During the talk of Chris, we have the opportunity via Q&A function of Zoom, as well as the chat function, to post questions. And after Chris' talk, we have the opportunity to discuss, and I will moderate the discussion together with Ellie. So Chris, I'm really looking forward to your exciting talk.
Well, thank you so much, Bernhard, for that very nice introduction. And what I'm going to talk to you about today as the lead off for molecular cuisine is my very personal and somewhat meandering path of discovery in psoriasis research over a considerable period of time. Let's begin with molecular cuisine. So what I'm going to do is to tell you how I started, which was a very rudimentary form of cooking over a single pot over a campfire, moving to what psoriasis research is nowadays, which is molecular cuisine, which for those of you who don't know what that is a modern type of cooking which takes advantage of technical innovations from scientific disciplines. So it's a very apt title for the way that most big team science is being done nowadays, and particularly in psoriasis. But throughout this talk, I want you just to bear in mind that however much we know about underlying pathomechanisms of psoriasis, and therefore finding new ways of treating the disease, we need to come back all the time to understanding those people that we are treating, our patients, because that is really the key to this research that I'm going to discuss, is actually the holistic approach. So this is where my research started. It's a long time ago now, 1983. I started in dermatology at St. Mary's Hospital in London as a first-year trainee. And in those days, this was one of the main treatments we used. It's still used nowadays, coal tar. We still don't know quite how it works, but it is not really a 21st century treatment. But also when I started in 1983, there were still two theories as to how psoriasis commenced. And these were put forward over 100 years ago by Radcliffe Crocker, a British dermatologist. And he said there are two theories as to how psoriasis commences. Either inflammation in the dermis is a primary event and hyperplasia is secondary, or inflammatory changes in the dermis are a secondary event. And in 1983, the vast majority of dermatologists, if not all of them, believed that number two was the correct one and that the immune system had nothing to do with psoriasis. Like most research, serendipity is important, and to be in the right place at the right time. And I was in the right place at the right time. I was working with Lionel Fry, Professor Lionel Fry, Professor Helgi Valdemarsen from Reykjavik, and their postdoctoral research, Barbara Baker, on the very early studies to show that psoriasis was indeed determined or mediated by T cells. And my main role at that time was to see the patients, to do skin biopsies, and keep the clinical material flowing to the lab. But this first paper, the conclusion at the end of that was that this work, which was looking at the influx of T cells into psoriasis during the evolution of the disease and then the efflux and the treatment, this showed that added further weight to the notion that the pathogenesis of psoriasis is primarily dependent on activities released by T helper cells. And we said activities, because in those days we did not know what the chemicals were or the signals were produced by T cells. Of course, now we know that they are cytokines, and but in those days cytokines were really very poorly understood. And that led then to this, the T cell mediated hypothesis of psoriasis, where CD4 T cells predominated in the dermis, and in the epidermis, as one might expect with an HLA-C disease, were predominantly CD8 T cells. But the problem was that most people around the world did not believe this. And then the other bit of serendipity had occurred, in fact, about 3 or 4 years before our work, where two Swiss rheumatologists, Müller and Hermann, had been using very high doses of cyclosporin, 14 milligrams per kilogram, to treat arthritis. And 2 of their patients had psoriatic arthritis, and their psoriasis completely cleared. And so that was the observation that cyclosporin would work in psoriasis, but their conclusion was wrong because they felt it was then an indication that psoriasis should be used in other proliferative skin diseases. But we were aware that cyclosporin was indeed a sort of rudimentary T cell immunosuppressant, because otherwise, why would it work in transplantation, prevention of rejection? So we then decided to do a study with low-dose cyclosporin for psoriasis. If it worked, that would prove the hypothesis. If it didn't work, well, we'd have to go back to the drawing board. It did work. And the conclusion from that paper, which published in the BMJ, was that the effectiveness of cyclosporin in psoriasis supports the hypothesis that psoriasis is a T cell mediated disorder. And it was that original observation that has led inexorably to where we are today with the biologic era for psoriasis. And also, importantly, was a long-term collaboration with industry, because if Sandoz hadn't decided to go with it and test our hypothesis with us with cyclosporin and then invest in the immunology of psoriasis, we would not have made the progress we have. I then moved to the University of Michigan in 1987 to work under the tutelage of John Voorhees, and I stayed there for just over 6 years. Whilst I was there, there was I worked with Professor Brian Nickloff, who's professor of pathology, the man here in the middle with the moustache. And we also at that time, there were 2 other illustrious future academics who were in that same laboratory. Jonathan Barker, who was with us for a year, and Frank Nestle, when he was actually a medical student visiting from Europe, also worked with us. But I was very lucky. Brian Nickloff at that time was just starting to do the original work of adhesion molecules in the skin. So we were able to show in the original studies that adhesion molecules such as ICAM1 are strongly expressed on epidermal keratinocytes. And that expression is induced to a major part by gamma interferon and to a lesser extent by tumor necrosis factor alpha. And the LFA1 on lymphocytes and the ICAM1 expression on endothelium and keratinocytes was the key reason that lymphocytes were trafficking to the skin and then trafficking to the epidermis in a disease such as psoriasis. So this is a sort of very simplified view of what we found, is that in keratinocytes and psoriasis, there's a induced expression of ICAM1, and on the endothelium, there's induced expression, or upregulation rather, of ICAM1 and induced expression of two other adhesion molecules, VCAM1 and E-selectin. So we've now moved on from showing that T cells were important, the work in Michigan, in St. Mary's in London, to now showing that they trafficked into the skin and they bound using LFA1. My next move was 1994, where I was headhunted to come to Manchester to set up the first academic department of dermatology in that city, and that's where I still am. Now, when I arrived in Manchester, there were three key initiatives that I introduced in 1994, which really paved the way for our psoriasis research. First of all, with my colleague Robert Chalmers, I established a dedicated psoriasis clinic, which is one of the first in the world. From that has come a number of studies, which I won't talk about today, but particularly around the introduction of psychology into management of psoriasis patients, and also work on the brain-skin axis. But this clinic has been the fulcrum or the source of all of our psoriasis research over the subsequent quarter century. At the same time, I established a dermatopharmacology unit, a clinical trials unit, which primarily was researching new treatments for psoriasis. And then the third thing which I did, which is important in this context, is that I initiated long-term collaborations with industry. So this is our psoriasis service as it was in the early days when we won a few awards for this. This was one of the first to be set up in the UK and certainly in Europe, and is now the largest in the UK. And interestingly, the first biologics that we actually trialed were those that targeted T cells. So Lefosept, which was never approved in the United Kingdom, but was improved in other countries around the world, which targeted CD2 and LFA3, and then efilizumab, Raptiva, which was eventually withdrawn, actually targeted those LFA1, ICAM1 that Brian Nikolov and I first described back in 1989. And that was a successful treatment, but most T cell targeted therapies don't work as well as anti-cytokines. We have trialed and investigated all of the biologic therapies which have been introduced over the last 15, 16 years into the NHS, and this has resulted in long-term, high-profile papers. And but as part of that, and a part of the introduction of these biologic therapies, came two key questions. What is the safety of these powerful cytokine-targeted drugs? And what is their long-term efficacy and durability of response? And to answer those two questions, but primarily the first one, I was involved in setting up a pharmacovigilance registry for psoriasis in the UK called BADBIR. Originally, the British Association of Dermatologists Biologic Interventions Register, and now because of small molecules, we've kept the acronym but changed the name to Biologics and Immunomodulators. I am still the chief investigator for that. We registered the first patient in 2007. We now have 165 dermatology centres across the UK and Ireland. And as you see here, we have close to 20,000 patients registered in this register on BADBIR, of which over 13,000 are on biologics. And we have a conventional cohort of patients on methotrexate and cyclosporin and other drugs, of about 6,000 patients. So we're learning a lot about these conventional therapies as well. But at the same time, in collaboration with Professor Catherine Smith and Jonathan Barker at King's College London, we have an aligned bioresource, which has over 7,000 of these patients who have DNA and or sera. So this suddenly becomes a very powerful resource for not just pharmacovigilance, but for stratified medicine. Now, those of you who've sort of been following the field of psoriasis and biologics will say, well, why do you need to really investigate it further? We've got great biologics. But actually, the answer is that we do need to do further work. Biologics are only used for severe disease, for whom it's been transformational for those patients. But there's variability in response. Not all patients respond, and not all patients have a long-term response. So that brings us to the next big area of my research as it's now progressed, which is stratified medicine. And Hippocrates, like in most medical conundra, got there ahead of us. And if he said it's far more important to know what person the disease has than what disease the person has. And that really brings forward the sort of personalized approach and understanding how different people respond to different drugs. So this is stratified medicine. So if I had one biologic to give to psoriasis patients, I wouldn't know presently who was going to be a responder, who was not going to respond, and who are going to be the few, but the important few, who are going to have serious adverse events. So it's very much a trial and error process at the moment. And what we're going to try and do is to make it a much more personalized and targeted approach. And this is where bad beer came in, because if we hadn't had bad beer in the bioresource, we would not have been able to accept the challenge for the Medical Research Council and David Cameron, when he was Prime Minister in 2011, to make the case for psoriasis being a stratifiable disease through this collaboration called SORT, Psoriasis Stratification to Optimize Relevant Therapy. And this is an industry academic collaboration. You see here the industry partners are not just pharmaceutical companies, but also diagnostic companies. And the academic partners, led from myself in Manchester, along with Richard Warren, my colleague, but with Nick Reynolds in Newcastle, and also Jonathan Barker and Catherine Smith at King's College London, and initially also with Frank Nestle. But it's but we're not just dealing with dermatologists. There are other scientists there as well: health economists, machine learners, bioinformaticians, immunologists, geneticists. This is true molecular cuisine, bringing in all the different disciplines, and including the patient group, to find which is the right biologic for the individual patient the first time round, and to try and take the trial and error out of it. So we've moved on from those 3 or 4 people who were writing those papers back in 1983, 84 on T-cell mediation, to just the 2 of us, Brian Nickloff and myself in the late 1980s. So now any paper has about 30 people on it, because all of their contributions are important. I'm just going to show you one bit of data from SORT. Ustekinumab is a biologic that targets interleukin-12 and interleukin-23. And we've been able to show that through the bad beer and the bioresource, that those patients who are HLA-CW6 positive have a much greater chance of gaining a 90% improvement in their psoriasis, the PASI 90, compared with those who are HLA-CW6 negative. That is not on its own a biomarker, but it could be put into the algorithmic mix, where we're trying to find what is the constellation of clinical, immunological, genetic, transcriptomic biomarkers, which can be put into an algorithm to predict and reproducibly stratify the response to biologics. But this is very complicated. And this multi-omic response needs to be looked at in 2 ways. Drug endotypes, where we're trying to find out what is the underlying algorithm that will allow us to say this patient will respond better to an anti-TNF rather than an anti-IL-17. But also, I think what we really are going to find is that we can start to reclassify psoriasis according to a molecular taxonomy, rather than the rather old-fashioned clinical phenotyping. But this multi-omic layering of platforms is tremendously complicated. And to be able to integrate that, is a good paper here written by one of my colleagues, Amy Fowkes, along with the SORT team, as to how we start to put all this multi-omic data together. Now, I've talked a lot about quantitative research, but it's important to view qualitative research. The National Institute for Health Research funded me for a program for over 6 years to look at the identification and management of psoriasis-associated comorbidity, the so-called IMPACT program. And this is because psoriasis is not merely affecting the skin. It also has significant medical conditions which are associated with it, such as psoriatic arthritis, cardiovascular disease, and depression. And therefore, you need to have a different perspective. So this is the IMPACT team. The only member of that team who's been in a basic science laboratory is myself. These are psychologists, health economists, and other scientists who are understanding the qualitative side of research and understanding and talking to the patients. So that work led to what we would call a new approach to the management of psoriasis, because it's complex, it's lifelong, and it's potentially life-ruining. And it has significant physical, psychological, and social impacts. So you need to research and to manage the patient according to all these constellation of other associated conditions. So it's not just about finding the right medicine, which we'll do hopefully with the SORT program, but it's also getting the patients to change their lifestyle. A lot of psoriasis patients have unhealthy habits. They're overweight, they smoke, they drink too much. Reducing all of those on their own can improve psoriasis, but combined with the right medicine is very powerful. But the only way to change people's behavior is by involving psychologists and behavior medicine experts in motivational interviewing. So that makes it much more of a three-dimensional approach. One of the big programs that we're running in Manchester now that has spun out of the IMPACT program is that we realized that really in secondary care, we were seeing psoriasis patients very late in their disease course. And we know from arthritis that the earlier the patient is seen after they've had a diagnosis of psoriasis, the more malleable it is to treatment, the more opportunity there is to actually reverse a lot of the changes and to prevent patients developing that lifetime cumulative damage, such as with psoriatic arthritis and cardiovascular disease, and not attaining their educational potential. So we've set this up. We're now seeing patients we've not seen before who have a different immunology because they're very early in the disease. And one of the big questions is whether we can intervene early on, maybe with the biologic therapies such as anti-IL-23p19s to maybe even reverse the disease altogether. The medical practice is moving from the older style reactive, where we're treating the disease, to one which I'm introducing here with stratified medicine, which is proactive and preventive, getting there early on. And that is the foundation for the Skin Science Foundation, which was launched by the ESDR and with industry partners at the Bordeaux ESDR meeting in September last year. And the premise of this is that we can ascertain what are the key discriminators of normal skin from diseased skin and intervene early. So to close, of course, I've talked about psoriasis really in the Western world and talked about biology, but psoriasis is a global disease. And the WHO in their report about 5 years ago challenged us to really do better as far as the epidemiology of psoriasis is concerned. And by so doing, really map out the disease burden and to allow any patient who around the world who has psoriasis to have access to the best quality care. And this is the underlying premise of the Global Psoriasis Atlas, which I guess is my last big program, last big project. It's now been running for 4 years. I direct this, and we've published our first major paper from that, which was a systematic review published in the BMJ just 2 months ago. And you can see here that we only have robust data from 1 in 5 countries around the world. There's a long way to go. But by building this data, by understanding comorbidities, understanding the health economic pressures of psoriasis on the country, we can maybe get patients, the governments of those countries, to actually invest more in psoriasis care and then benefit even more patients with psoriasis. So to close, my last slide is my sort of view of the systems holistic approach. Now, I'm doing this lecture today, not from Manchester, but from London. I'm going to use London as the example. I've talked to you about the importance of understanding the patient, but also understanding the systems biology. So maybe a systems biology person or an immunologist would think of London in this way. This is a map of the underground, and he or she could easily navigate their way around London. They could get from Heathrow to Oxford Circus, or from Westminster to Oxford Circus, just by using this map. And this could well be the circuitry for transcriptomics or immunology, but they would have no understanding, unless they'd come up above ground, as to what great city London is. And this is the qualitative side. This is the other side of the research, which we're trying to fit together to see that, okay, you might understand how it all works, but how does it affect the workings of London itself? So hopefully what I've shown you over the last 20 minutes is that it's been an interesting and fascinating journey. There's a lot of research I've not discussed with you. And also there's a lot of questions which still need to be answered. And in a way, I wish I was starting again. But thank you for listening.
So thank you very much, Chris, for this great talk and impressive journey in the area of psoriasis. So I may kick off the first question, and I would also like to ask everybody to post questions at the Q&A area or at the chat function. So Chris, could you hypothesize, or if you have evidence, why is psoriasis so important for the human being that it is conserved all over the world in a relatively high prevalence and in a similar prevalence in the different areas?
Yeah, well, that's an excellent question. Obviously, I didn't have time to go into the genetics in any way, but it does have a high prevalence. And that means there has to be some evolutionary survival aspect to this. And I think that the current theory, which I think is probably the best one, is that in the pre-antibiotic era, streptococcal infection was a significant, serious problem. And it seems that probably patients with psoriasis may have in some way have been immune or less or likely to be carriers of streptococcus rather than succumbing to it. And therefore that was the evolutionary advantage. Okay, you're going to have this tough skin disease called psoriasis, but at the same time, you're going to be able to survive overwhelming infection. And that, of course, is beneficial both to the host and also to the organism. I think that is probably the most likely reason. Anything that's common probably has the evolutionary advantage to it at the time. In the post-antibiotic era, of course, it's not such an important protective mechanism.
Right. Just to add on this, if we know that the TH17 and IL-23 pathway is important, and knowing that yeast infection are controlled by this, we do not, from our current perspective, yeast infection is not a major problem, but somehow psoriatic inflammation is an exaggeration of this archetypic immunological response. Mm-hmm. Do you think for early human beings, this type of immune system was more important?
Well, I think it is. I mean, I think in most places around the world, fungal infection is actually a major problem. We don't see it so much in Europe nowadays, but certainly in the work that we've done in looking at patients with psoriasis in Africa and elsewhere, the tinea corporis is without doubt one of the commonest skin diseases, but also deep fungal infection as well.
So we have two questions right now I would like to pose to you. There's one from Elisaveta Gripaleva on potential biomarkers that could predict the response of patients to biologics. If you could give us some insights from your perspective, what could help us already now?
Well, I mentioned the work on HLA-CW6, and certainly, those that are patients who are positive are more likely to respond to ustekinumab, and conversely, those who are negative are more likely to have a better chance of getting a good response to adalimumab. But that's not all patients. It's going to be part of that algorithm. We know that there are clinical features, which I would say as a biomarker. So people who are overweight, less likely to respond well. Women don't respond so well to biologics. Patients with psoriasis on the palms and soles. But so that's just one other facet which you can then put into that model. So it's not a simple one biomarker. And my oncology colleagues don't like me saying this, but psoriasis is a far more complex disease. Although we've learned a lot from oncology, I just know that to come up with a single biomarker is just not going to be viable in psoriasis. It's going to be a complex algorithm.
So there is another question from Nicholas Haas, asking whether there are other species that develop psoriasis naturally.
No, as far as we know, there's no other species that develop psoriasis naturally. the animal models are all models which have been bred in that, the classic one is the imiquimod mouse model, but there's no other animal model that we know of that has psoriasis.
Yeah, another one is from Marie Losset. Your thoughts on using preventive therapies such as statins among patients with moderate to severe psoriasis to reduce cardiovascular risk?
Well, that is an excellent question. And so I think that the use of statins, there's two parts to that. There have been some studies, small studies a few years ago, showing that statins, which do have some anti-inflammatory effects, may actually be able to improve psoriasis itself. But I agree that with the high risk of cardiovascular disease, particularly in those patients who have concomitant psoriatic arthritis, the use of statins early on, or certainly the targeted screening of patients with psoriasis for high cholesterol, and we've done this in Manchester and picked up a lot of cases. And in that early access clinic that I mentioned, about 40% of the patients we've seen had at least one cardiovascular risk factor, and particularly high cholesterol. So I think that may well be an interesting approach.
And there is another question on disease modification and potential markers for identifying patients at risk to develop psoriasis. And what are your thoughts to how long will it take that we get the biomarkers to identify patients at risk and intervene? What would be your
Well, it goes back to the other question. I think that if we can treat patients early, that's going to make a huge difference. And if biologics, and now with biosimilars, we have the anti-TNF biosimilars at a tenth of the price they used to be. But I think that what we now know about tissue-resident memory T cells, and that's one of the reasons that they persist in the areas where psoriasis has previously been, and that's why psoriasis relapses in the same sites. If we could maybe intervene very early on and prevent that permanence of tissue-resident memory T cells, we might be able to reverse the whole process. But that's speculative. So I think it's treating early, it's addressing the risk factors for some of these diseases through screening, and still searching for biomarkers, or probably complexes of biomarkers which will allow prediction.
So there's one additional interesting question from Linda Grine, and she asks you, to what other disease can we translate our knowledge on psoriasis, and what would be your advice how to follow this in this context?
Well, that's a nice question, actually. And what I'd like to believe is that the work that we've done with psoriasis and the sort of the various processes we've gone through with the registry and the SORT program are a good model for other chronic inflammatory diseases. And I know that the people who are working on atopic dermatitis have certainly been able to evolve and accelerate their programs of work much more quickly than they would have done, I think, if we hadn't been the pathfinders, if you like, in psoriasis. I'm not saying it's easier. I think atopic eczema is an even more difficult disease, but at least we've sort of, in a way, shown how to do it. And this is, I think, a good blueprint or template for any disease, any skin disease. And it's all around collaboration. One of the things I didn't emphasize enough was how important the collaboration is, not just with like-minded individuals who are dermatologists, but also bringing in those other aspects of the cooking, the health economists, the immunologists, et cetera, to really look at this in a three-dimensional way.
Yeah, there is a question on management advice. With all your achievements, Nicole Ward is asking, how do you recruit the right people? How do you mentor? And how do you achieve such a staggering success with so many people? So
that's a nice question from Nicole. And I think it's recruiting people who are curious and are inquisitive and want to understand. They don't just go in the clinic and just treat patients and that's it. They think, well, why is it that stress has made this patient's psoriasis worse? You know, it's those questions that you take from the clinic and then try and address with modern technologies. And if you don't have that inquiring mind, and you find that out very quickly, people working with you, if they have it or they don't, then they probably aren't going to make a lot of progress. And I like people who are willing to push back as well and not accept the dogma, which is how I started in 1983. Lionel Fry did not accept that psoriasis was not an immune-mediated disease and set out to prove that it was, and he was right.
Yeah, always stay hungry.
Always.
So thank you very much, Bernhard.
Running.
And thank you very much, Chris, for this really brilliant lecture. I think it gave us the true dimension of what research means and what research can give. And just in closing, just a brief glimpse at what is awaiting us in 2 weeks from now, from the recipes of a master chef to new master chefs. So in our freshly baked session, we will be hearing about high impact papers. Next speakers will be Cindy Irwin and Wim de Cler. This will be a really wonderful lecture. We are really looking forward to hear about these very new and exciting new findings. We have a lot of other exciting events ahead of us, but most importantly, we would like very much all of you attendees and those who have not been with us today, we really would like to welcome any suggestion on your hand of interesting topics to discuss in any of our 4 series. So please be in touch and let us know. See you in 2 weeks. Until then, stay well. And thank you very much for having been with us. Goodbye.
Thank you.
Goodbye, and thank you, Chris.
Okay, pleasure.
Thank you.